Hello students, this is the first practical of Amphibian lab. It is a lab in which experiments were done on the frog, which are not done nowadays. You have to just learn about the results of those experiments as well as few things about the instruments that those were used. So in this practical we will just study the instrument appliances which were used in the Amphibian lab one by one. You have to remember one thing, you have to write down the full name of any instrument. For example, here it is a Starling's Chymograph. So instead of writing just Chymograph, you have to write down Starling's Chymograph. Okay, so that thing you have to remember. Now about the instrument. This instrument is used for the recording the movements of muscle which is wrapped over the cylinder, black color cylinder. So the movements of skeletal muscle or any other muscles will be recorded over a graph which is wrapped over the cylinder, and the cylinder is being moving. Okay, so for that chymograph is used. Now various parts, as you can see in the figure, the red arrow shows the cylinder over which this graph has been wrapped. Then the blue arrow shows the spindle by which this cylinder has been linked with the motor. Then this yellow arrow shows contact box, and the green arrow shows the speed limiter by which you can increase or decrease the speed by changing the gears of the cylinder. And the black arrow shows leveling screw by which you can just level this chymograph. Now behind this contact box shown by the yellow arrow, there are contact arms which are linked with the spindle. So whenever this cylinder moves, these contact arms will be also moving. Now when they just come in contact with the contact box, they will make a circuit. So these contact arms can be used to make or break the circuit. So with each revolution, these contact arms will make contact with the contact box, and this in contact box the circuit will be made and break. Okay, so there are such two contact arms, so you can make or break the circuit twice in each evolution. Okay, so that is the use of contact arms. And also you can change the speed of the cylinder based on your need. Okay, so that's it about the Starling's chymograph. Next instrument is Myograph board, which is a wax topped wooden piece with Myograph stand on which this Myograph board has been fixed. Now Myograph board is used mostly to fix the tissue preparation with the help of pins on the wax. It has a Myograph stand which is triangular heavyweight steel base on which this Myograph board has been put. Okay, now Mercury key. Now it is known as Mercury key because small cups shown by the red arrow on the right side are filled with the mercury. Mercury is a liquid metal which is filled inside those cups. Now when mercury is filled and when you put this handle downwards, these two steel hooks which are seen in the diagram will make contact with the mercury. So now these two steel hooks are connected with each other. So circuit will be completed. Circuit wires has been joined with these two contacts shown by the green arrow. So when you connect circuits with these two contacts, and when you put this handle down, circuit will be completed, and when you put this handle upwards, circuit will be broken. So This is a mercury key which is used to make or break the circuit. Okay, the next instrument is Dubois-Raymond induction coil. Now this instrument is used basically to convert galvanic current into the Faraday current, or direct current into the indirect current or induced current. It is basically a step transformer used to obtain high voltage stimulus from a low voltage direct current source by using principle of Faraday's electromagnetic induction. As you can see in the figure, it consists of mainly two coils, primary coil and secondary coil. Primary coil is fixed with the frame, and secondary coil is movable. Now here it can be moved only horizontally away from the primary coil, but in some advanced induction coil it can be moved at an angle of the primary coil. So primary coil is consists of 300 turns of insulated thick copper wire around a soft iron core, while the secondary coil consists of 5000 turns of very fine copper wire. And as you can see, there is no contact between the primary coil and secondary coil, physical contact. So current is not passed from the primary in the secondary directly. But what happens when current has been passed in primary coil via primary circuit, then it will generate a magnetic field which will cause generation or induce current in secondary coil. So this magnetic field will cause generation of the current in secondary coil, which will be passed to the secondary circuit. There is no direct physical contact between these two coils. Now you can increase or decrease the strength of the induced current by two mechanisms. One is, I told you, by distance between these two coils. When you increase the distance, strength will decrease. And second is by changing the angle between the two coils. In some advanced induction coils, you can change the angle between the primary and secondary. So by changing angle, you can also increase or decrease the strength of the induced current in secondary coil. So this is how strength of the stimuli has been increased or decreased. Now the third part, as you can see in the figure, is Knipf's hammer. Now Knipf's hammer basically used to make or break the circuit, but very rapidly, almost 40 per second. So it is used to give us stimuli for the making or breaking the circuit. So it is nothing to do with the conversion from galvanic to Faraday, but this is just used to make or break the circuit. Okay, so that's it. This one is a Detainer set. It consists of a stand on which a metal strip has been mounted, and a second stand on which a mercury cup has been mounted, in which mercury has been filled. As you can see in the figure, this metal strip, when is put on vibration, will go up and down. When it goes down, it will dip inside the mercury cup and will cause making of circuit. When it will goes up, it will cause breaking of circuit. So when it is put on vibration, it will rapidly make or break the circuit. So it is used to make or break the circuit rapidly when you want to generate tetanus in a muscle, skeletal muscle. So same as Knipf's hammer, it is also used for the making or breaking circuit very rapidly, but not as rapidly as Knipf's hammer, which can make or break circuit 40 times per second. This is less than that, and you can change the frequency of the stimuli by changing the length of this metal strip. So when you increase the length, stimuli numbers, frequency of the stimuli will decrease, but when you make this metal strip smaller, it will give more stimuli per second while generating tetanus in skeletal muscle. Okay, next one is time tracing tuning fork. Now as you can see, there are two types of tuning fork in this figure. Upper one is clinical tuning fork and lower one is time tracing tuning fork. I have put these two because you just confuse in these two types of tuning fork. Lower one is time tracing. Now there are some differences between these two. One is there is a white marker attached with the time tracing, but there is no such marker in clinical tuning fork, that is upper one. Second difference is the frequency of the tuning fork, which is 100 in case of time tracing tuning fork, but in case of clinical tuning fork it will be either 128 or 256 or 512. In this figure it is 256. Third difference is a base. There is no base in time tracing tuning fork, but there is a base which is marked by the green circle in clinical tuning fork. So base is present in clinical, but there is no base in case of time tracing. And the fourth difference is the length of the arms. Length of the arms are slightly longer in case of 100 Hz, but it is smaller in case of 256 Hz. So that will change depend on the frequency. So apart from that, there are three differences by which you can differentiate these two types of tuning forks, clinical or time tracing one. This white marker is used for the time tracing. Now where it is used, it is, as the name suggests, it is used for the tracing the time. Now on the right side, as you can see in the graph, there are some waves made at the bottom of the graph marked by the green arrow. These waves has been made with the help of this time tracing tuning fork. So when this tuning fork has been put on the vibration, it will make such waves over the moving Myograph. Okay, so these waves will help to find out various periods over the graphs, because this one wave will be made in 0.01 second based on the frequency of the tuning fork, that is 100 Hz in this case. So 100 such waves will be made in 1 second. 100 Hz means 100 times vibration per second. So 100 such waves will be made in 1 second. So you can divide 1 second in 100 parts. Because these periods are very smaller, you can measure with the help of this time tracing tuning fork. Okay, next one is writing lever. Writing lever mainly are used to magnify and record the muscle contraction or the smoke paper wrapped over the moving cylinder of Myograph. So there are so many types of writing levers: simple lever, afterload lever, frontal lever, muscle lever, heart lever, but mostly we use two types of lever. One is muscle lever, that is shown in the left diagram, and heart lever, that is shown in the right diagram. Now let's discuss various parts. As you can see in the figure, the part shown by the red arrow is adjusting screw, by which you can just fix this writing lever or the muscle draw or Myograph board. Then the part shown by the green arrow is a afterload screw. Then part shown by the black arrow is a straw, and by the yellow arrow is a stylet or marker. The same stylet or marker you have seen in the time tracing tuning fork as well. By using this, we can make waves over the smoke paper. Apart from that, you can see some holes in left strip. These holes are used for the hanging various weights, which will be used to see the effects of free load and afterload over the skeletal muscle. Okay, so these are the various parts. Few parts will be less in heart lever because there is no need to hang weights in heart lever. So heart lever is slightly simple in nature. Okay, so right one is a heart lever, which is used to see the movements of a heart, frog heart, and the left one muscle lever is used to record movements of skeletal muscle of the frog. Okay, so these are the writing levers. Okay, next one is a pulse commutator. This is used to change the direction of a current. It consists of a vulcanized base on which a rocking metabolic cradle is mounted, and there are 6 cup-like depressions as you have seen in Mercury key. So it is also you can call it as a modified Mercury key, acting as a two-way switch. Okay, so now this 6 cup will be filled with the mercury, like a Mercury key, and 6 terminals are attached to these cups. So as you can see in the figure, this metallic cradle, which is in the center, can be moved over the left side, which is left in the left diagram, and in the right side or the right diagram. Now when you put in the left side, it will divert the current toward the one tissue, and when you put it on the right side, it will divert the current towards the another tissue. So when you want to just alternate the current from one tissue to another tissue, you can use this pulse commutator. Now next one is a simple electrode. It is a very simple instrument made up of 2 copper wires mounted on a non-conducting material. This is mainly used to convey Faraday current from the secondary coil to the living tissues. So that means to apply stimuli. So when you want to apply a stimuli to any living tissue like a muscle or nerve, this instrument is used. It is known as simple electrode or stimulating electrode. Okay, stimuli. Stimuli is a change in external atmosphere that will provoke response in any excitable tissue. Now stimuli can be either mechanical, thermal or chemical, electrical or osmotic. Excitable tissue can be either a muscle, skeletal muscle, cardiac muscle, smooth muscle, nerve or gland, etc., etc. So if you want to excite or stimulate this excitable tissue, for example skeletal muscle, you have to apply a stimulus, whether it is inside the body or outside the body. So for example, if our brain wants to contract any skeletal muscle, it will have to just give a stimuli first of all to that skeletal muscle, which is basically action potential, which is electrical in nature. Then some hormones can give stimuli to some glands to give a response, which is chemical stimuli. Hormones are chemicals, so that is chemical stimuli. Action potential are electrical in nature, so that is electrical stimuli. So these are the examples of stimuli in our body. But when you dissect out a skeletal muscle of a frog and then put on the myograph board and just want to record the contraction of the skeletal muscle, you have to give stimuli. Now the nerve is not connected to the brain of a frog, so you have to give a stimuli in another form, that is electrical current. So we will give electrical stimuli, but which is not in the form of action potential, and it will be in the form of electrical current. Now we will use electrical stimuli mostly because there are some advantages of this electrical stimuli in comparison to other stimuli. Those are electrical stimuli. It is very easy to deliver, operator can handle it conveniently, and the apparatus which is required for delivering is also very tidy. Then apart from that, it is very easy to grade the electrical stimuli for the desired strength, frequency and duration. If you want to give 1 millivolt current, you can give. If you want to give 2 millivolt current, you can give, which is not easy to grade mechanical stimuli or thermal. So electrical stimuli is very easy to grade. Now advantage of that is also it will be very least injurious. By chance you will not give large stimuli. Okay, so chances of injury will also decrease. Now apart from that, stimulus is very easily controlled by making or breaking the circuit. Then stimulus is very accurately localized also, and stimulus can be controlled from the longer distance as well. So these are the various advantages of electrical stimuli, and that's why we will just use electrical stimuli in the form of electrical current in comparison to others. Okay, so we'll just put a simple electrode. I have told you as well at that time, a simple electrode will be used for delivering this electrical stimuli to the excitable tissue, whether it is nerve or skeletal muscle. So if you want to contract a skeletal muscle, we'll just make a contact of simple electrode to that muscle or nerve connecting to that muscle, and then we'll make a circuit and we'll give desired stimuli by just increasing or decreasing the strength and desired numbers of stimuli. If you want to give 5 stimuli, you can. If you want to give 10 stimuli, you can. So you can grade them. You can grade in the form of strength or frequency or duration. Okay, so we'll give desired strength of stimuli with desired frequency to that nerve, and muscle will contract, and we will record the contraction by putting simple electrode over the excitable tissue. Okay, so that's how we'll give a stimuli. Now as I have told you, this stimuli will be given by using circuits. To give electrical stimuli, we'll use circuit. Now circuit is of two forms, primary circuit and secondary circuit, as you can see in the figure. Primary circuit means which have a source of current. Current can be either direct or indirect. Now first of all we'll use direct current, but we can't give this direct current to the tissue because we can't grade this direct current directly. So what we'll do, we'll just convert this direct current into indirect current first of all by using induction coil. As I have told you, Du Bois-Reymond induction coil is used as shown by the red arrow in this circuit. Now this induction coil will be connected to both primary circuit as well as secondary circuit, as you can see in the figure. This induction coil is present in both the circuit, primary as well as secondary. In the lower portion it is present in primary circuit, and in the upper portion it is present in the secondary circuit. Now primary circuit will be connected to the one part of this induction coil, and lower part will be connected to the secondary circuit. But this there will be no contact between the primary circuit and secondary circuit inside this induction coil. So when the primary circuit is completed, it will cause generation of the magnetic field in the secondary coil, and then secondary coil will generate the electrical current inside the secondary circuit. And at the end of secondary circuit, you can see there is an electrode which will be connected to the excitable tissue, which can be nerve or muscle. Okay, and then it will be the stimuli will be delivered. So whenever there is a need of any other types of key like tap key which is present in primary circuit, or you can also join Neve-Sammer or Paul's commutator to change the tissue, you can connect in primary or secondary circuit. So that is all about the circuits, primary as well as secondary. One thing that you have to remember, that primary and secondary circuit are not connected with each other. There is no physical contact between the primary circuit and secondary circuit. Primary circuit will will have source of electrical current, and secondary circuit will just deliver that stimuli to the tissue. That's it. Okay, that is everything about the instrument. Those are used in lab 1, that is Amphibian lab, and some of the introductory aspect of the Amphibian lab as well. Thank you.